In the last module, we learned how genes encode proteins through the processes of transcription and translation. What we didn't say was that this is a finely tuned process, and proteins are only made when they're needed. In other words, gene expression is regulated. Before we talk about regulation, let's first define gene expression. Gene expression means making proteins based on the information in DNA. This is a familiar topic for you because we've covered it plenty, which brings us, once again, to the central dogma of molecular biology. DNA encodes RNA, that's transcription, and RNA encodes protein, that's translation. In other words, there's a flow of information from DNA to RNA to protein. DNA contains the information necessary to make all the proteins that you need. When a protein is made, we say that the gene is expressed, or we could say that the protein is expressed. Proteins in turn carry out all of an organism's functions. The genes that an organism expresses determines an organism's ability to function in its environment. That environment can change over time, necessitating a change in gene expression. Regulation of gene expression, then, determines how much of a given protein is made at any one time. In the simplest description, making a protein includes transcription and translation. We know, however, that each of those steps have many sub-steps, and that there are other steps as well, especially in eukaryotes. The most important point of regulation is transcription, especially in prokaryotes. As we go along, we'll see why that's the case. It's also true in eukaryotes, but transcription and its regulation are more complex in eukaryotes, and there are more steps overall in gene expression, and each of those steps is subject to regulation. Here are some examples of regulation of gene expression. Prokaryotes are continuously exposed to environmental changes, to which they have to continuously adapt, and that means changing gene expression. Here, E. coli, which normally live in animals' large intestine, are growing on a petri dish, a very different environment than the human gut. At other times, it will find itself in wastewater or a variety of other environments, which vary widely in terms of temperature, nutrient content, salt concentration, etc. E. coli has to adapt to these different environments in order to survive and reproduce. It adapts by regulating gene expression. We'll look at two specific examples. In the presence of the sugar lactose, E. coli will turn on genes necessary to metabolize and derive energy from lactose. And that's the term we use, by the way. We say that genes are on, actively being transcribed, or off, not being transcribed. Of course, it's more nuanced than that. There are degrees. A gene can be off, it can be on at a low level, on at a high level, and everything in between. For the sake of simplicity, however, we often say a gene is on or off. Another example we'll look at is tryptophan production. E. coli behaves the opposite with regard to tryptophan. In the presence of tryptophan, it will turn off the genes necessary for tryptophan production. These are just two examples. There are many more. In high salt concentrations, for example, E. coli turns on genes that help it cope with the additional salt. At high temperatures, E. coli will turn on genes to protect proteins from denaturing. Multicellular organisms, like mammals, are different. We don't cover physiology in this class, but a central aspect of physiology is homeostasis, the maintenance of a constant internal environment. Internally, your body maintains a constant temperature, a constant blood pressure, constant nutrient levels in the blood, and many other parameters. Mammalian cells, therefore, don't have to respond to a constantly changing environment like E. coli do. The degree of regulation of gene expression is much less than it is in E. coli, with the exceptions of certain cells. There is, however, an important aspect of gene expression in mammalian cells that is highly regulated: tissue-specific gene expression. Every cell in your body has the same genome. Recall that multicellular organisms begin as a single cell, a zygote. It divides to make the trillions of cells of an adult organism. All of those cells have the same genome that was present in the original zygote. Despite this fact, there are many different types of cells. That's what this diagram is showing. If we take an eye, for example, we can see several different cell types. Cells of the cornea, for example, are different than cells of the vitreous, which are different than cells of the conjunctiva. There are several more cell types in the eye. We could also look at other internal organisms. Cells of the liver are different than cells of the gallbladder or the intestine or the pancreas. There are hundreds of different cell types in the human body. What makes one type of cell different from another, and how does the body make all these different cells if they have the same genome? The answer is that one cell is different than another because of the genes it expresses. Every different kind of cell expresses a different subset of the genes in the genome. As we'll see, there are two main causes of tissue-specific gene expression: epigenetics and transcription factors.
Now we come back to a lesson we had very early on in the semester: the differences between prokaryotic and eukaryotic cells. Prokaryotic cells, you'll recall, are simpler, lacking a nucleus or other internal organelles. There are fewer steps in gene expression, and its regulation is simpler, occurring mostly at the level of transcription. Eukaryotic cells, however, are more complex. Gene expression is more complex, and there are more opportunities for regulation. Most importantly, the nucleus separates the steps of gene expression. Transcription happens in the nucleus, and translation happens in the cytoplasm, and these processes can be regulated independently. There's more to it than that, though. Eukaryotic cells have large linear chromosomes as compared to the smaller circular chromosomes of prokaryotes. They're also wrapped around proteins called histones and are highly compacted in cells. This affords another important level of regulation. How tightly compacted the chromatin structure is dictates which genes can be expressed. This level of regulation is often called epigenetics, which we'll explore further a little later on. Recall also that mRNAs are processed in eukaryotic cells, which includes splicing, which is also a regulated step. Nuclear export of mRNA and mRNA stability in the cytoplasm are also regulated. The upshot is that transcription is the most important level of regulation in both prokaryotes and eukaryotes. But gene expression in eukaryotes is more complex, involving extra steps, and all those steps are potential sites of regulation. We'll begin with a classic example of gene regulation in bacteria, the lac operon. When I say classic, I really mean classic. Jacques Monod and François Jacob won the Nobel Prize in Physiology or Medicine in 1965 for elucidating the lac operon. I learned it when I was in college, and now you're learning it. Lactose is a sugar present in milk and elsewhere. Sugars, as you know, are a source of energy for humans and for bacteria. Lactose is a disaccharide, and the first step in burning lactose for energy is to break it down to its component glucose and galactose, which is accomplished by the enzyme beta-galactosidase. You know what happens from there. Glucose enters cellular respiration, starting with glycolysis, to produce ATP. Galactose can be converted to glucose and also enters glycolysis. Lactose metabolism is regulated, and we know this because of data like that shown in panel B. In this experiment, bacteria are grown in the presence of glucose and lactose. The growth of the bacteria is shown along the y-axis, and time is shown along the x-axis. Initially, growth is rapid, but then plateaus before beginning rapid growth again, a biphasic growth curve. Marked along the x-axis is the fuel being consumed. The bacteria are initially consuming glucose almost exclusively, and then later on they start to consume lactose. This graph tells us two things. First, the bacteria prefer glucose as a fuel source. If it's available, they will only consume glucose. Second, when the bacteria switch to consuming lactose, there is a lag in growth, represented by the plateau. As we'll see, this plateau represents the time it takes to turn on the genes necessary to break down lactose, and these genes only turn on when glucose is exhausted. This phenomenon of switching fuel sources is called dioxic shift. Before we can discuss the gene regulation underlying dioxic shift, we have to learn a little bit about the organization of genes in prokaryotes, which can be different than eukaryotes. Many genes in bacteria are organized into operons, a group of related genes plus their regulatory elements that are regulated as a single unit. Let's take that one at a time. The lac operon contains a set of related genes called LAC-Z, LAC-Y, and LAC-A, which all play a role in lactose metabolism. LAC-Z, for example, encodes the enzyme beta-galactosidase, which breaks down lactose into its component monosaccharides, glucose and galactose. LAC-Y and LAC-A also encode proteins that are involved in the use of lactose that I won't go into here. The operon produces a single mRNA that encodes all three proteins. Consider the efficiency of this arrangement for a moment. All three proteins are needed to use lactose, so they're all regulated together. That doesn't happen in eukaryotic cells. The other part of the operon is the controlling elements. In this context, the word element means a DNA sequence to which proteins bind to regulate transcription. The controlling elements here are the promoter and the operator. We've already seen the promoter. In bacteria, it is the sequence to which RNA polymerase binds to start transcription. Note that in the case of the LAC operon, a sequence called the operator separates the promoter from the protein-coding genes, LAC-Z, LAC-Y, and LAC-A. The operator is the binding site for a regulatory protein called LAC repressor. As you might guess from its name, LAC repressor represses or turns off the LAC operon. When repressor is bound to the operator, it blocks progress of RNA polymerase and there is no transcription. The sugar lactose binds to and inhibits the LAC repressor. If it's inhibited, that means it's not bound to DNA and RNA polymerase can make RNA normally. Lactose therefore induces the genes necessary for its own metabolism. Let's go over that again. The LAC operon is inducible. That is, it can be turned on in the presence of lactose. First, lactose is absent. Repressor is bound to operator, transcription is blocked, and the lactose metabolism genes are off. Second, lactose is present and binds to the repressor, which inactivates it, meaning that it no longer binds to the operator. RNA polymerase is now free to do its job, which is to transcribe genes, and the lactose metabolism genes are on. RNA is made and the proteins necessary for lactose metabolism are made, including beta-galactosidase, which cleaves lactose so it can be burned for energy. Let's pause for a moment and appreciate the simple elegance and efficiency of this system. When lactose is present, the proteins that the cell needs to metabolize lactose are made and the cell will use lactose as an energy source. When lactose is not present, the cell simply doesn't waste time and energy by making these unnecessary proteins. Or to state it as I did before, lactose induces the genes necessary for its own metabolism.
Now let's look at a contrasting case, the trip operon, which is a repressible operon. That is, it is normally on, but is turned off in the presence of the amino acid tryptophan. Let's think for a moment about why that's the case. The trip operon contains the genes necessary to make tryptophan from scratch. Tryptophan is an amino acid, which the cell constantly needs because the cells are always making protein. To fill this need, the trip operon is normally on, and the cell can make all the tryptophan it needs. If, however, tryptophan is available in the environment, the human gut for E. coli, which can be rich in tryptophan, then the cell can turn off these genes and save itself the time and energy necessary to make tryptophan as long as it's present in the environment. Let's see how the trip operon is repressed. Note that the trip operon has a similar organization to the LAC operon. You have a series of structural genes, which encode the enzymes that make tryptophan, and upstream of that, you have the promoter. Between the promoter and structural genes, you have the operator sequence once again. The normal state is shown at the bottom. Normally, the trip repressor is not bound. RNA polymerase is free to transcribe, and the enzymes for tryptophan synthesis are made. The repressed case is shown at the top. If tryptophan is present, it activates the trip repressor, meaning that it will bind DNA at the operator and block RNA polymerase, and the operon is turned off. The proteins needed to make tryptophan are not being made, and the cell is not making tryptophan. Rather, it's getting tryptophan from the environment. This is the opposite of the LAC operon. Lactose turns on, induces the LAC operon, and tryptophan turns off, represses the trip operon. While the trip operon is repressible and the lac operon is inducible, note that they are both built on repressors. That's a brief explanation of regulation of gene expression, how it differs between prokaryotes and eukaryotes, and two classic examples from bacteria. In the next video, we'll take a deeper dive into regulation of gene expression in eukaryotes.